So I'll get this recorded. Okay, so yeah, the next part of the biological molecules topic for A-level biology is enzymes. So we're sort of building on the knowledge of the proteins that we did before and actually looking specifically into enzymes. So enzymes are a protein. And so we're going to look at how they work, their specificity, what a biological catalyst is, how they work, look at activation energy, and look at how they work as enzymes inside the human body. So as we know from GCSE, enzymes are specific and they can only work with certain substrates. So we have this, originally we had this lock and key theory, which we'll look at again, but we'll also look at a new theory onto how enzymes work as well. But it's all that idea that they have a specific shape. So like you can see on the image, almost a jigsaw puzzle pattern, that only specific substrates can react with enzymes. So enzymes can either put two substrates together and build them into a larger molecule, or they can break substrates down to make them into smaller molecules. So enzymes. Enzymes are proteins that are called biological catalysts, and they are able to bind to substrates. And so what they do is they're able to speed up a reaction. All catalysts, so catalysts you'll come across in chemistry, are anything that speeds up a reaction. But what's really special about enzymes as being a biological catalyst is they don't get used up. So for example, a catalyst in a chemical reaction that's not a biological catalyst can get used up. So we can add catalysts to reactions, but they can get used up. So we have to replenish them, restock them. But for an enzyme, they don't get used up. They are continually recycled. They're really, really specific, like we said before. So one particular enzyme will act as a catalyst for one reaction only, and it can't be used for multiple different types of reactions, can't be used for different types of substrates. And so what these biological catalysts do is they speed up a reaction without being used, and they can act inside or outside of a cell. So digestive enzymes are extracellular enzymes. So these are enzymes that work outside of the cell. These are enzymes that break up macromolecules into micromolecules. So they're breaking up those large molecules, those large food groups that we're eating, things like carbohydrates, and we were breaking them down to something smaller, breaking them down to the smaller substances of things like glucose. So they're an extracellular enzyme. But we also have enzymes which control reactions inside our cells, which we call intracellular enzymes. So we'll look at a lot of mixtures of different types of enzymes that do both, and they come up a lot. Lots of enzymes are involved in controlling replication of DNA. So they would all be intracellular enzymes, and then we'll have like extracellular enzymes. So enzymes as well, if we're thinking some are extracellular, some are intracellular, they're going to be quite a broad variety of shapes and sizes as well. A good tip, enzymes, their names usually end in ASE, in A's. So for example, some that you've come across before, some extracellular ones that are digestive enzymes are things like carbohydrates. So these change carbohydrates into sugars, lipase, lipids into fatty acids and glycerol, and then protease, proteins into amino acids. So it's breaking up the substrates into the new composites. So enzyme keywords. So they are biological organic catalysts, and the compound that the enzyme acts upon is called the substrate. So whether it's an enzyme that's building two substrates together to make a larger molecule to bind them together, or it's breaking a substrate apart, it doesn't matter which way around that reaction is happening. We always call the compound that reacts with the enzyme a substrate. And then we have that part of the enzyme that we've talked about loads when you've done GCSE, and that is the surface onto which the substrate binds, and we call that the active site. So that's the part that has that specific shape that only allows a specific enzyme and a specific substrate to react and bind together. So because of that, each enzyme has a particular shape, and when our substrate and enzyme join together and bind, we call that an enzyme substrate complex. And our energy required to make a reaction is our activation energy. So enzymes. Enzymes are globular proteins that act to speed up chemical reactions inside organisms. So if it's a globular protein, these are proteins that are going to have a quaternary structure. So we're talking about complex proteins here made up of multiple polypeptide chains. They can make reactions occur at least 1 million times faster than they would normally occur. So some of these reactions just wouldn't be feasibly possible in the human body to take place without enzymes being there. For example, digestion, our body might eventually be able to break down some of those foods, those macromolecules that we eat, into macromolecules, but it would take a lot of physical breaking down. It would take a lot of time, a lot of effort, a lot of energy. So enzymes reduce that amount of energy that's needed to break down those macromolecules. They have that very, very precise 3D shape that we've spoken about with quaternary structure proteins before. And because it's got a 3D shape, it's held into this precise structure with those bonds that form between the amino acids in the chains. So things like the disulfide bridge bonds, the hydrogen bonds, the ionic bonds, the hydrogen bonds, all of those are all taking place to hold this enzyme into place. And they always have a depression of the surface of the molecule, and that is our active site. So a depression is an area that concaves inwards, and that is our active site, the bit that has that specific shape that allows a specific substrate to bind with it. So each enzyme has a specific shape and works with a specific substrate. And you'll start to see that I'm using the word specific a lot. This is one of those words that when you get to writing exam questions and answers, if the word specific isn't included, you don't get a mark. So if you said enzymes have a shape and this works, it allows a specific substrate to bind, you would need to actually say enzymes have a specific shape, that active site has a specific shape, which works with a specific substrate. So you need to have that specificity defined in there. And again, like I've said, they can split up a substrate into smaller molecules, or they can join substrates up to make them into larger molecules. So lock and key theory. There's two models of enzyme action and how enzymes work at A level. We have the original lock and key theory, which is what is covered at GCSE, and then we have another theory. So we'll look at lock and key, we'll reintroduce that again. So the idea behind lock and key theory is that the shape of our enzyme's active site is complementary to the shape of the substrate or substrates, if it's joining more than one together, that fit inside it. So when the substrate moves into the active site, the enzyme substrate complex is formed. So it's this structure, this idea. So we have our substrate here. This is our enzyme. This is its depression here, this area. So this is its active site, this whole area. So that shape of the active site is specific to the shape of the substrate. They match like a lock and a key. So the idea is that, for example, you can only fit one. A lock has to match the key. So a key can't just go in any lock, it won't unlock any lock. It's the same idea between an enzyme and a substrate. So when they join together, they get the enzyme substrate complex, and the bonds in the substrate are weakened. For example, in this one, they're being broken apart, and then we have our products. You can also get the other way around, but that's lock and key theory. They automatically have these specific shapes. The enzyme has a set shape, the substrate has a set shape. They join together, they either break apart the substrate or the substrates bound together, and we have our products. Also now have induced fit theory. So lock and key actually works on the idea that they are perfect. They fit perfectly together, nothing needs to change, and it will just automatically go into shape. Induced fit theory actually looks into, well, is it going to definitely always be that exact shape? So induced fit theory suggests that enzymes actually change their shape slightly when the substrate enters the active site. So what happens is when that enzyme and the substrate comes together, so for example here, we've got our active sites present on this enzyme, and we've got our substrates here. We can see that they are of a similar shape, but they are not exactly the same. When that substrate comes together with the enzyme, we have this shift in the enzyme structure. And then what you can see here on the enzyme substrate complex is the enzymes now have actually changed shape slightly as the substrate has bound. And so then it binds to form the enzyme substrate complex. And when it binds to form this enzyme substrate complex, the active site has actually slightly changed shape. So we have our enzyme product complex now. So we've made our products. So it was in this instance the splitting of these two. We have our products being made, and then they're released, and then our active site returns to how it was originally. It doesn't stay in this changed format. So the shape change helps the substrate to fit more tightly. It's the idea that actually, if they're just going to go in and fit in at the exact shape, it might not actually be perfect. If we're having lots coming in, lots coming out, it might get damaged. So somebody said, well, actually, no, maybe it changes, and then it changes back, and it changes, and it changes back. This is now the more accepted theory. It's the theory that people go with, actually, that it's induced fit. When you look at it, it's very, very similar to lock and key. It's just with this actual idea that it has a similar shape, and then it changes to allow it to fit exactly. It's still a specific enzyme for a specific substrate. It's still a specific shape active site, but it's just that the shape changes ever so slightly to accommodate that substrate even more, so it fits even more perfectly, which is the idea that was more believable than lock and key, where we're saying that they just automatically all have the perfect shape. So a question that often comes up in exams is things like, describe the key differences between lock and key theory and induced fit theory. So for something like this, you would want to highlight the differences in the two theories. So for example, I'm going to just annotate on here really quickly. Describe the key differences between lock and key theory and induced fit. Lock and key, and I've just put L and K, we have no change in active site shape. And that is the key difference. There is no change in the active site shape. Whereas in induced fit theory, we have a slight change once we our substrate and enzymes start to bind, the active site changes slightly, and that allows it to have that closer, snugger fit for when they react together. So what is activation energy? So we sort of started off this whole thing of, okay, we have enzymes, they're a quaternary protein, they are a biological catalyst. Biological catalyst speeds up reactions. They speed up reactions by lowering activation energy. And activation energy is often this term that's thrown around a lot. We never really clearly define it. So activation energy. Activation energy is the energy that is required in order for a chemical reaction to take place. So that is how much energy is needed for that chemical reaction to take place. So when we are breaking down those macromolecules to micromolecules, it is a chemical reaction. When we are forming polypeptide chains, when we're making, that isn't a chemical reaction because we're doing different bonding. But in this situation, when we're breaking things apart using enzymes, we're having a chemical reaction. So how does this link to enzymes? Well, what happens is activation energy. Energy is needed to convert substrates into products. So we need that energy to convert those substrates into products. So for example, if we use the example of carbohydrates, we want to break them down into glucose. So we want to break them down into their monomers. We need energy to do that. We have all those really, really strong bonds holding them together, and we need to break them. To break them, that requires energy. So the energy required to start a reaction we call this the activation energy. So this is the energy that allows that reaction to take place. Otherwise, it would just be happening all the time, which wouldn't be very efficient in our body. So activation energy is the amount of energy that we need, the minimum amount of energy that we need for that reaction to start. So activation energy can be provided by increasing temperature, but in most living organisms, raising temperature is not an option. So if we just raise the temperature in our bodies, we're just going to get hotter and hotter and hotter, and basically eventually cells would end up dehydrating, and organisms would end up dying. It just wouldn't work. It's not a useful way for us to reach activation energy. Some chemical reactions that we would do, not in biology but in chemistry, yeah, you can actually just increase the temperature, increase the rate of reaction, because we've increased the activation energy, we've increased the temperature, the activation energy is reached. But in organisms, most of the time, that's not really an option. We can't actually do that. So activation energy, we're just supplying more energy. It's the amount of energy that's supplied to the molecules. So yeah, if we heat them up, they will have more energy. Can't do that to humans, can't do it to animals, can't do it to plants. There's other side effects of us doing that. So we have to look at something else. So enzymes come in and they reduce the activation energy needed for a reaction to start. And so what I'm going to talk through here are these graphs. These graphs appear all over the place. So what we have is we have our activation energy that's needed. I'm going to actually possibly draw one. So this blue line here is our activation energy with an enzyme. So we're actually going to ignore that for a second. We're going to look at this pink line. So this pink line, where we have this increase in energy, so where this arrow is here, activation energy without enzyme, that is how much energy, this increase from here to here is how much energy is required for that reaction to take place. So we have the amount of amount of energy required for the reaction to take place, and then the amount of energy reduces to then the amount of energy that's required for then the products to be produced. Enzymes will actually reduce that activation energy. So then if we look at the blue line, that's the activation energy that we need if enzymes are present. So we don't need as much energy. Enzymes actually lower that activation energy. Again, we've got another example here. Normal reaction, that's the amount of activation energy that would be required. Enzyme catalyzed reaction, that is how much activation energy is required. So there's a huge substantial difference between the two. So enzymes, what they do is they lower the activation energy needed for a reaction, and they can do it in one of two ways. So what they can do is, when we're looking at the active sites of an enzyme, the charged areas of that active site and the substrate attract. So again, it's looking at those attraction forces that we've talked about before with bonds. And what can happen is the charge areas of the active site and the substrate attract each other. And what this does is this causes the substrate to be held in a position that encourages bonds to be made or broken. So actually, by holding it in place, the enzyme's holding the substrate in place. And if we're going to form bonds, we're then holding the two substrates in a position that means that they are more likely, it's going to encourage bonds to be formed because we're going to have those right charges or the right attractions being faced towards each other. In a similar way, it can do the opposite, but if we're wanting to break bonds, it holds them in a way that we have forces of repulsion going and it will want to break apart. Another way is our active site could contain amino acids. Now I'm using the word could, not all will, because some will work in the top way. If some enzymes are working in the top route, they don't need amino acids to be present. However, if they're working in this bottom way, some will have amino acids present. Because one enzyme only works for one specific reaction, we'll never have a crossover of the two for one enzyme. So the active site might contain amino acids. And what's really good about amino acids is they have that acidic side chain. Acidic conditions help reactions to take place. That's another way we can increase the rate of reaction. If you think back to GCSE chemistry, you can increase rates of reactions by increasing the temperature, increasing the pH. Again, living organisms cannot just flood their body into acidic conditions to make reactions take place faster. It isn't going to work. An example where that does actually happen is the stomach. Our stomach is really acidic, has really highly acidic conditions. So actually reactions take place really, really quickly in our stomach, i.e. the breaking down of macromolecules to micromolecules. We still have enzymes in there that then speed up reaction even more. So we actually have like double whammy in our stomachs. But the whole of our body wouldn't be able to consist of the acidic conditions that our stomachs are. So then we will also have enzymes that have these amino acids at their active site, which actually increases this acidic conditions, which helps the reaction to take place, which is really, really clever. So you start to see lots of these graphs, and what you sort of have to know about them, it's not too much that we have to know, but basically we have our amount of energy that's needed, and we have our energy of our reactants, and then our energy of our products. So our activation energy is the amount of energy that's required from where we started off, what is the original energy, free amount of energy we started off with, and the increase of the energy. So enzyme catalyzed reactions. The speed of an enzyme catalyzed reaction, we can measure it in two possible ways. We can either measure it at either the rate at which our substrate is used up. So for example, if we were making, if we are breaking down carbohydrate starch into glucose, and the rate at which our carbohydrates are used up. Or we can measure it the rate at which the product is made, so we can actually measure it how quickly is our glucose being formed. So we can measure it in either way. So either the rate at which our substrate is used up or the rate at which our product is made. So our speed of our enzyme catalyzed reactions can be altered by changing a couple of things. We could change our enzyme concentration. So we could increase the amount of enzymes present to increase the rate of reaction. The more enzymes that are present, the more sites of reaction of presence and more active sites of presence, and the more substrates combined, the more reactions can take place. We can also speed up by increasing the amount of substrate present. Obviously, if we don't have much substrate, our reactions aren't going to take place really quickly. The more substrate we have, the faster it will take place. The temperature, like we said before, increased temperature, rate of reaction is going to increase. Not necessarily the best thing in living organisms. And pH, if we change the pH, make it more acidic, our rate of reaction will increase. Again, not always applicable in a living organism, but our enzymes have found a way around that. They actually are going to increase the pH by having a slightly, acidic active site, or being present in places like the stomach, which have slightly more, highly more acidic conditions. So enzyme catalyzed reactions, we can increase the substrate concentration, and you have to sort of get quite used to some of these graphs that you start to see, and things like points of saturation. So what you get is you have basically a rate of reaction graph here, and we're saying the rate of reaction will increase, so there's a positive correlation as we increase the substrate concentration until where X is, and that is what we call our point of saturation. And this is where increasing our concentration does not affect our reaction rate. So by this point, X, we could keep chucking in more and more and more and more and more substrate into our reaction, into whatever we're wanting to happen. So let's say we were breaking down starch in a beaker to produce glucose, doing that. And we can just keep adding in all our substrates, but it will not, the rate of reaction will only increase to a certain point. We have this point of saturation. what often happens is you'll get exam questions that will say, suggest why increasing the concentration of the substrates does not increase the rate of reaction past the point of saturation. Well, what we have to think of here is that actually enzymes and substrates are working in a partnership. We will get to a point that no matter how many substrates we start chucking into a beaker, we only have a set amount of enzymes. Now, those enzymes don't get used up, they can get reused again, but the reaction can only happen faster whilst there's free active sites. Once your amount of substrate matches your amount of enzymes, you're then going to eventually get to a point where our rate of reaction can't get any quicker because we have no free active sites. All the enzymes are being used up at the moment, so our rate of reaction cannot increase any quicker. So a point of saturation would be that all our enzymes are being used and we're having to wait for an enzyme to be free for the next substrate to come in and be used. So that was what would be happening at that point of saturation. Again, we have another similar graph for if we increase the enzyme concentration, but this time we don't have a point of saturation. Now, you can get this graph sometimes you get a different graph. So this one would only be true. Oh, let me go back. This one would only be true if we had a completely infinite amount of substrate. So if we had an infinite amount of substrate going into a reaction, what could happen is the more enzymes we kept adding in, the faster and faster and faster and faster this rate of reaction would go. However, you could reach a point of saturation if we only have a set amount of substrate. So actually, there's no point just chucking in like the world's largest amount of enzymes if we only have a certain amount of substrate. So what you'd actually end up with, again, is if we had a set amount of substrate, it would be a limiting factor, and we'd actually end up with probably a graph that looked a bit like before. We'd have a point of saturation, and that would be to do with the amount of substrate present. So that's another thing that can affect enzyme catalyzed reactions. Don't want that bit yet. I've lost the bit I want. So things that can affect enzyme activity. So we've said temperature is one. So I'm just going to get up a blank screen actually. I want to discard that. I've lost a screen that I wanted. So let's add on a blank screen. New slide. Okay, and we'll have a look at what factors can affect enzyme activity. So temperature was one that we spoke about. So obviously we know temperature can affect rates of reactions, but temperature can actually affect enzyme activity as well, which makes sense. It's a biological molecule, and so temperature can affect that as well. So the idea is that temperature, any increase of temperature is going to increase the kinetic energy of the molecules present. So temperature will increase our kinetic energy of the molecules. So that makes sense as to why our rate of reactions will increase. Oops, molecules. If we increase the temperature, because our kinetic energy is increased, therefore our activation energy will increase. So temperature increase creates, this kinetic energy of molecules to increase. If our kinetic energy of our molecules have increased, we're going to have an increase in collisions. So we'll then get an increase in collisions. And by increasing collisions, we're talking about increase of collisions of molecules. So we have increase in collisions. Increase in collisions. the idea is that actually if we increase the temperature of something by 10 degrees, the rate of reaction can actually increase by approximately 2 times. So it can double. So every 10 degrees that we increase temperature by, the rate of reaction doubles. Which would seem like, yeah, that's an okay thing. Like we can keep this, we're happy with this. However, that thing that we've heard about a lot, denaturing, comes into play. So we cannot just keep increasing temperature when enzymes are in play. So what will happen is if we just keep increasing the temperature when we're dealing with enzymes, what will happen is our enzymes active site will begin to denature. And what we mean by denature is its active site actually changes shape. So denaturing is when actually those bonds are being broken. So if we think about an enzyme, it's a protein, it's a highly complex 3D shape that's held together by lots of complex bonds. We know one thing that breaks bonds is temperature. So if we increase the temperature too much, our enzymes are going to break. So actually, regardless of whether we can break down our substrates, we're actually going to cause our enzymes to start to change shape, because that really, really specialized folding that's in place that makes that active site starts to break down, because we've gotten rid of lots of those bonds due to that increase in temperature. So the enzyme denatures. So denaturing is officially the changing of the active site shape. And that is a massive, massive issue with temperature. So we can't just increase temperature over time. We can't just lower temperature over time. So a graph that would show that is, so for temperature, the rate of reaction, when we're dealing with an enzyme is, it'd be, if we put temp here, and we're going to put rate of reaction over here, it would increase until the optimum, and then it would decrease again. Now, we've actually got this bit going to 0. You sometimes might see it a little bit differently, where it actually might look more like this. Enzymes denature when it is too hot. The increase in heat is what breaks down the bonds. So denaturing occurs because bonds are broken, which changes the active site. When we decrease the temperature, bonds aren't broken. Decreasing the temperature doesn't break any bonds in the enzymes, but it's going to reduce the amount of energy. So it's going to then be harder, even with enzymes, to reach that activation energy. Our collisions have reduced. So actually our substrate's actually getting to our enzymes, that has reduced. So everything has slowed down. So it's just going, everything's going to take longer. So our rate of reaction is decreased with a decrease in temperature, but it's not actually to do with denaturing. Denaturing is only when the temperature is high. So this is at high temperatures. And when I say high temperatures, I mean anything above the optimum. So denaturing is only involved in enzymes in temperature when we are talking about above the optimum. So that's our optimum. This whole range here, rate of reaction is decreasing here because enzymes are denaturing. If an enzyme is denatured, it basically means we've broken it, we've ruined it, it's changed, it's not functional, it cannot do its job. So then actually we have to wait for the body to make new enzymes for them to then carry out the job. So it's why just somebody getting a slightly high temperature when they're poorly can create massive issues, is because suddenly we have lots of enzymes denaturing, we don't have enough enzymes to carry out basic biological reactions. Decrease in temperature, again, we still have a decrease in rate of reaction, so that is still dangerous, but actually it doesn't affect the integrity and the structure of the enzymes. So if we can then get that body temperature back up in a safe and controlled manner, it's better, because those enzymes can get back to work again straight away. We're not waiting for new enzymes to be produced. So that's to the benefit there. what we then have is pH. So let's have a talk about pH. pH works a little bit differently. So we've said acidic conditions increase rates of reactions. Well, how does pH actually affect our enzymes? So enzymes in the human body, their optimum temperature is 37.5 degrees. That is our temperature of our body, internal conditions of our body. So anything above or below, the enzymes aren't going to be working at optimum. Different regions of our body have different pHs. So for example, our stomach has a pH of 2. So the optimum for an enzyme that we would find in our stomach, like pepsin, would work at pH 2. So it would be something that looked a little bit like this on our pH scale. The rest of our body, for example, might have an enzyme that's working in the small intestine, which is going to be a pH of 7. It would look more like this. So that would be our pH of 7 under there. That's our pH of 2. So our optimum has just changed location. So that's quite important actually to remember that pH, enzymes have different optimum pHs. So they have different optimum, have different optimum pHs depending on their location in the body. And what organism they're found in actually. So differing optimum pH due to location. So now we're going to look at actually what pH does. So again, we're actually going back to this idea of enzymes. They're a protein, they're a complex protein, they're a 3D protein. They're held together by all these bonds. What do? Well, what ends up happening, I'm going to actually type this on rather than, is we're going to look at hydroxide and hydrogen ions. So pH creates a change. So if we have changes in pH, that creates a change in the number of hydroxide and hydrogen ions that surround the enzyme. So if we have a change in pH, as you know about acids and alkalis, that means we have a different number of hydroxide and hydrogen ions around our enzyme. What this does is these interact with charges on the enzymes. So if we have different numbers of hydroxide and hydrogen ions, these ions, what they will do is they interact with the charges, on the enzyme's amino acids. So we're now talking about those enzymes that have amino acids on them. So they interact with changes on the enzyme's amino acids. So what will happen is, if we're talking about enzymes that have got amino acids at the active site, we've already said, oh yeah, these amino acids on the active site, they're really, really good. They actually increase the rate of reaction. Yeah, but if the pH changes that we find those enzymes in, those amino acids are going to be actually affected. So what will happen is those ions that interact with the charges on the enzyme's amino acids, they affect the hydrogen and ionic bonding. So actually we're starting to affect the whole structure and formation of our protein again. So this will affect our hydrogen and ionic bonding. If our hydrogen and ionic bonding is affected, you can probably guess where this is going. It will cause changes to the tertiary structure. Oh, let's just type tertiary. Tertiary structure of our protein, which in this case, the specific protein we're talking about is an enzyme. So that will change shape, which will mean our active site will change shape, which basically means it will denature. pH too acidic, it's going to denature. pH2 alkali is going to denature, and it's because of that amount of hydrogen and hydroxide ions around the outside. It's just going to affect those hydrogen and ionic bonds that are taking place at the enzyme. Now, and that is a big difference between pH and temperature when it comes to talking about whether enzymes denature or not. Is temperature, enzymes only denature when the temperature is above the optimum. When it's pH, well, actually, the second that the pH starts deviating from the optimum, it either becomes too high or too low, then actually the enzyme can start to denature straight away. So that is sort of a rough idea of what happens with pH. That's the biggest difference, really, with pH and temperature of enzymes. I just want to pop back, actually, and just look at... Oh, no. Okay, right. They're all drawings. This point again. So when we were talking about enzyme catalyzed reactions, and I've said the substrate increases. So if our substrate increases, that means that we've increased our amount of molecules able to form enzyme substrate complexes. So because of that, our initial rate of reaction will increase. When all enzymes are in use, we cannot increase that point anymore. So our point of saturation is when all enzymes are in use. And it's just another point that I wanted to, it's the same thing again, but I wanted to re-go over because, again, this comes up a lot in exams and it's often a misunderstood point. You'll get a graph like this and it says, what's caused this point of saturation? Suggest. And I'll often use the word suggest, and actually it's not a suggest. Well, you are suggesting something, but you know from enzyme theory that obviously this point of saturation has occurred because all our enzymes are in use. Similar, we have the enzyme catalyst reaction. So enzymes increases the rate of reaction to catalyze in a given time, and it will just keep increasing if we're talking about a given time with an infinite supply of substrate. If it has not got an infinite amount of substrate, what will happen is it will do this, and we would actually have rate of reaction, and if we had it over time, for example, rather than over enzyme concentration, rate of reaction over time, it would increase and then it would just stop. And it would just stop because then no more reactions can take place. So that's another thing to sort of make clear. So sometimes it can be rate of reaction against enzyme concentration on the graph. Sometimes it could be rate of reaction over time. And then it says, why is it just increasing with a positive correlation? Well, it's increasing with a positive correlation because actually we're just continually increasing the amounts of enzymes, and it will continue to increase the rate of reaction until we use up all substrate, until the reaction is complete. So the last little bit about enzymes is enzymes can work in a couple of different ways, and we can have inhibition of enzymes as well. So because enzymes are a biological catalyst, it means they're not used up after a reaction. So they're still in the human body. They're still floating around in the human body, waiting to be used again. That can present a problem. It can actually present an issue that actually sometimes we don't want the enzymes to be used right here, right now. We don't really have necessarily what you might call like a storage place that we like hide all the enzymes in and then we release them when we need them. We do release them from places. So for example, like the pancreas, but once they're released, it's sort of a bit more complicated. By the time they're getting recycled and sent back to where they've come from, they could carry on doing reactions. So actually, we have something called inhibitors, and we can inhibit enzyme activity as well. So sometimes we might want to switch enzymes on, might want to switch them off, and might want to do that quite quickly. So we have a couple of different ways we can actually inhibit enzymes from working. And the first way is through competitive inhibitors. So this is an example of competitive inhibitor. So we've got enzyme A, its substrates come along and it cannot bind at the active site because there is an inhibitor in place. So what happens is competitive inhibitors are chemicals that will either slow down or completely stop a chemical reaction. So why it's slow down or stop is because we may not have an inhibitor inside every single enzyme at every single active site. So some substrates may still be able to bind, whilst other enzymes have inhibitors in them that stops the substrates from binding. So these are chemicals. So the inhibitors are chemicals that are either going to slow down or stop the chemical reaction completely. And they do this by preventing an enzyme substrate complex being formed. So they either cannot be formed at all, or they are occurring at a lower rate. So again, when we're saying it's occurring at a lower rate, it's still physically stopping them happening in other enzymes, and it will just be that some enzymes don't have the inhibitor in. So it causes a reaction to happen at a low rate because a majority of the enzymes have these inhibitors in place, which are stopping the chemical reaction taking place. What we find about them is they are similar in shape to the normal substrate. So what they just do is they go in and they block the active site. So they have a very similar shape to that substrate. So we're talking again about that specificity of the shape of the active site of a substrate. Increasing substrate concentration can compensate for the effects of this one. Now, what that means is if we increase how much substrate is present, it means that whilst, okay, yes, an inhibitor's in place, a substrate can't bind, it can't make an enzyme substrate complex, it can't go and make its product. There will be other enzymes that don't have the inhibitor in place. So those ones can take place, so they can happen quicker. Also, the inhibitor won't stay there forever, so it will have that sort of recycling process again as well. So what will happen is the longer that we have those inhibitors in place, the higher the concentration of a substrate, it can compensate for the effects of an inhibitor being present. Another way we can inhibit the use of enzymes is through non-competitive inhibitors. So non-competitive inhibitors have reversible effects. Some have reversible effect and others are irreversible and denature the enzymes. So I'll sort of talk about them slightly separately. So what they do is this time they bind at a different part of the enzyme. They don't go to the active site, and they will change the shape of the active site. This can be either reversible and it just changes it for a short period of time, or it can be permanent and it denatures the enzyme. So these are non-competitive inhibitors. They are not competing with the amount of substrates present. They go and actually bind onto the enzyme at a completely different point, and they are going to go and change the shape of the active site, either permanently or for a short period of time. And the last little bit we want to do is just have a little talk about enzyme controlled reactions. So rates of reactions. So basically, we just want optimal conditions all the time. Optimal conditions is the most important thing, really, when we're dealing with enzyme reactions. We just want optimal conditions constantly. Optimal conditions. So they are not, and that's a really, really important thing, they are not the same for every single enzyme. Every single enzyme has its own optimal conditions. You may find two enzymes that have the same optimal conditions, probably because they're found in the same part of the body where they work. However, not every enzyme will have the same. So for example, when I was talking about pepsin, which is found in the stomach, that has an optimal pH of 2. Lots of enzymes will denature at a pH of 2. So denaturation can occur if external conditions causes change to the tertiary structure, which alters the active site. So that's the main idea, that denaturation occurs when we change that tertiary structure. when that active site is changed. And the last little bit is actually calculating the rate of reaction. So let's have a little look at this. So calculating the rate of reaction. So the rate of reaction, we can either calculate it due to the increase in products over time, so increase in products, or we can calculate it due to the decrease in substrates over time, or reactants over time, depending which terminology you want to use. What we can do is when we have those graphs, we can actually calculate the gradient of a graph of an enzyme activity against the time, and that will give us the rate of reaction in AU, and AU being arbitrary units. So I'll just type that down as well. So if we're able to calculate the gradient of a graph of enzyme activity against time, that will give us rate of reaction in AU, and AU being arbitrary units. And that pretty much is everything for stop recording.